Let's apply what you've learned about plant cells and osmosis to a real-world situation. This case study will help you connect cellular water movement to something very familiar: French fries. Kevin tries to make French fries at home. He cuts russet potatoes into strips and fries them until they're dark brown, but instead of being crispy, they're soggy. And even though he cooked them long enough, the texture just isn't right. Kevin's brother suggests soaking the raw potato slices in salty water for about 15 minutes before frying them. He claims this will help the fries become crispy. Let's think about why this might work from a cell biology perspective. So why did the fries turn out soggy? Even though I'll give you a little help, make sure you record your answers in your lab notebook. Potatoes are made of living plant cells, and those cells contain a large amount of water. As the fries cook, that water must escape before the surface can become crisp. If too much water remains inside the tissue, it turns to steam and prevents browning and crisping. Go ahead and pause to note down your answer. What happens when potato cells are placed in salty water? Salty water is a hypertonic solution compared to the inside of potato cells. Through osmosis, water moves out of the cells and into the surrounding salt solution. This causes the cells to lose water and shrink slightly. Make sure you pause the video to write down your answer. How does this help make fries crispy? When the potato slices lose water before frying, there is less moisture inside the tissue. With less internal water, less steam forms during frying, allowing the surface temperature to rise high enough for browning and crisping reactions to occur. In other words, osmosis removes excess water before cooking, setting the stage for a crisp texture. Almost done. Go ahead and pause to note your answer. On the cellular level, the salt solution pulls water out of the potato cells. The central vacuoles shrink, turgor pressure decreases, and the tissue becomes less saturated with water. This is a perfect example of how osmosis directly affects food texture. This case study shows that osmosis is not just an abstract concept. It influences plant health, food texture, and everyday cooking results. The same principles you observed in Elodia and potato cells explain why salty water helps make French fries crispy. Kevin's soggy fries were the result of too much water trapped inside potato cells. Soaking in salty water creates a hypertonic environment that draws water out by osmosis. With less water inside the tissue, the fries can brown and crisp properly in hot oil. Once again, we see how cell structure and water movement directly affect real-world outcomes. Throughout this lab, you explored how plant cells are specialized depending on the role of the tissue in the plant. You observed protective cells in the onion and tomato epidermis, photosynthetic cells in Elodia, storage cells in potato tubers, pigment-containing cells in tomato pulp, and defensive structures in the purple heart stem. You also saw how different plastids, chloroplasts, ameloplasts, and chromoplasts are all related but specialized for different functions. The osmosis activity and case study demonstrated how water movement affects cell structure, turgor pressure, and even real-world outcomes like food texture. Finally, structures such as plasmodesmata and raphides show how plant cells communicate and defend themselves. Together, these activities reinforce a central theme in plant biology: structure supports function. From the microscopic level of organelles and membranes to the survival and success of the whole plant.